Hi, I'm Miss Coulter. I'm the Year 12 biology teacher here at Harris Academy Beckenham. In Year 12, we will study biological molecules, cells, respiration, photosynthesis, and DNA, and we cannot wait to have you start with us.
Hi, I'm Miss Moore. I'm the Year 13 biology teacher here. In Year 13, we study nervous coordination. Also, we look at genetic expression and genetic inheritance. And we also look at genetic engineering. And I cannot wait to see you in my Year 13 class.
Now, why study biology at A level? If you wish to pursue a career in biology, this is important. So further education courses in university, such as biomedical science, biochemistry, dentistry, medicine, pharmacology, all that you see there. Having an A level in biology is very useful. Some universities require you to. In addition to this, the skills that you learn from studying biology are valued by employers. Some students go on to follow degrees such as law or engineering after doing an A level in biology, as this is a highly sought after course. So how is A level biology different to GCSE? Now, this is going to be a lot harder than you remember. So the 3 exam papers that you sit at the end of the year at A level is what you're going to be tested on. Nothing else matters. Although the lab book does count, you won't be, as I said, marked on it, just tested for evidence for meeting those 5 skills. Now, the good news is you won't be given a grade that you don't deserve. You won't be given a grade that you don't deserve, but you'll need to understand, to get a good grade, you'll need to understand the specification and beyond very well. Now, majority of questions will be given in various contexts to test your knowledge. Now, you will be used to, at GCSE, to get a grade 9, that will only be 5% of the paper's marks. So at A level, 5% of the marks is for grade E only. So you'll need to go above and beyond the specification in order to achieve the best grades. You've never had to answer questions like this before. Now, in order to support you with this, in addition to feedback within lessons, you'll be encouraged to keep a summary book. Now, a summary book is not a redo of your notes that you do within lessons, but a way for you to revise so that you're not overwhelmed by the content when the exams come over. A lot of this content will be new to you, and to avoid cognitive overload, you'll need to do plenty of pre-reading and follow up after the lesson. Now, the summary book allows you to facilitate this. And this will be needed to be completed every week, and will include your notes in class in neat, as well as any further information or reading that you have done. There'll also be at least 3 not homework tasks, but individual study tasks, which I or another biology teacher will be setting for you to complete each week to direct you towards consolidating your knowledge. So a quick sample lesson, one of my favorite topics. So just have a look at this image here and what is it showing? You may pause the video here. So what you're seeing here is a technique known as somatic cell nuclear transfer, or cloning to the layman. Now, you may or may not have heard of Dolly the sheep. Dolly the sheep was the first animal to be cloned in a lab, and this was done in 1996, but was actually only announced in 1997. Now, Dolly was the name of the sheep that was cloned. So how Dolly was created was that Dolly's original, an egg cell was taken from a donor, and what we call a diploid cell from Dolly's original was taken. This is the nucleus from a complete cell. Now with the egg cell, the nucleus was removed and the nucleus from the diploid cell inserted. And this is done similar to how in vitro fertilization is carried out. Now that egg cell will then be matured in a petri dish until it forms a ball of cells called a blastocyst. Now this group of cells is essentially the clone. It has the same genetic material as the original. Once that blastocyst has matured, it may be implanted into a surrogate mother. Okay, maybe implanted into a surrogate mother, which case it will mature naturally within the surrogate mother until it is born. Now, Dolly may be the first mammal cloned by somatic cell nuclear transfer, but it wasn't easy. It took 277 attempts to do this. And we still have Dolly cloned, the clone of Dolly the clone in a museum. Although Dolly died prematurely due to arthritis, we don't know if clones suffer from any health problems. But since then, we've had several clones of other mammals, including Gcse the cat, or copycat, in 2002. Ralph the rat, also in 2002. Dewey the deer, cloned in 2003. Snuppy the puppy in 2005. And most recently, Zhongzhong and Huahua in 2018. Now, what does this mean? Does this lead to humans cloning eventually? Will we have a attack of the clone scenario? Well, not necessarily. Human clones are currently illegal and no one has attempted it. But what do you think? Should we be able to clone humans? Now, why should humans research this technology? Now, it may lead to producing animals, such as farm animals with desirable characteristics. Maybe cows that produce more milk, or sheep that produces more wool, etc. Rather than the selective breeding that humans have been doing for thousands of years, we can get a stable and quick breed or certain characteristics that we can get once in one generation. We can produce animals that be genetically engineered to produce human products. Now, we already have this, where we do this with bacteria, things such as insulin. But with cloning, we can mass produce other products, including things like spare livers. We can address problems with organ donors. And lastly, we can also clone human embryos potentially to provide stem cells, which are cells that can differentiate or turn into any cell type. So over to you. What do you think? Should we be allowed to clone humans? Do the advantages and the potential uses outweigh the ethical concerns or the things we don't know about? Are we allowed to do it? Should we be allowed to do this? Thank you for listening, and I'll hope to see you soon.